Hi everyone, welcome to this episode of Keipert Labs. In today's video we're going to be discussing amino acids as another family of compounds that we need to be familiar with, especially in a forensic context with their connections with proteins. Okay, so when we're looking at amino acids, we're looking at substances that have a really peculiar type of functional group, and they look like this. Okay, so what we have over here, if I'll pick a different color for a second, we know we've got, this is called a carboxylic acid group. And then over here, we have what's called an amine group. So it's called an amine group because it's related to ammonia. Now, we don't, in our course as it stands, that we don't look at amines in any more detail than this. There are a family of organic compounds that occur very frequently in nature, and they are basically basic compounds. And so what we, the reason that we're considering them here is that amino acids, these amazingly interesting naturally occurring compounds, have this functional group that is a combination of the amine and the acid group, hence amino acids. Okay, and because these two functional groups, or these two sections, work in tandem, that's why we consider them to be a whole separate family. They're not acids and they're not amines, they're a combination of the two. And then what we see is that actually, I didn't really need to do that yet. We see that this R group could represent just about anything. But now the reality is that these are naturally occurring substances, that we don't get an infinite number of naturally occurring combinations here. We get only a very limited number, about 20 or so naturally occurring amino acids, each one with a particular name and shorthand abbreviation for the biologists who may or may not be that, enjoy chemistry as much as we do. Okay, and so some of the, I'm going to draw you some of the common examples that we come across. Okay, so these are three examples of amino acids that we come across: glycine, alanine, and serine. Okay, that these have, biologists tend to give these a three-letter code, so that when we're starting to look at how amino acids combine together to form polypeptides, and which then fold together to form proteins, that then we can look at the sequence of these particular amino acids. So glycine is the simplest. We have just a hydrogen off the side. Alanine is a methyl group attached off the side. And then serine, we've got CH2OH attached off the side. Now, there are others that are more complicated. We're going to go through some of those different categories of functional groups in a minute. But what you can see is that for these three acids, that the functional group here is identical. So it's the side chain, we call it, which dictates the difference in how these amino acids behave. Because the differences in their side chains is what distinguishes them from one another. Okay, so we're going to have a quick look at some of those categories of side chains now. Okay, so when we're thinking about the nature of these side chains, there's two main categories that we would consider them to be in: non-polar and polar. Okay, so based on exactly kind of what interactions that we have here. Okay, so our non-polar example might be thinking about our alanine with the CH3. Okay, so that's non-polar. Okay, so our R groups, so mainly just carbons. It can be small like this, we can also get bigger. So phenylalanine involves, so would have our CH2 and then a benzene ring that looks like that off the side of our amino acid, so phenylalanine. So our definition, so a carbon-based side chain. It's a bit vague, I suppose, but it doesn't it doesn't have, so that it is just typically just carbons. There's no kind of other polar groups that are attached. And then out of the polar ones, then we can kind of consider them to be acidic basic, or non-ionic. Okay, that we might say. Okay, and so acidic, the side chain contains a COOH group, so one that's not part of the amino acid. Okay, so we've got one that's called aspartic acid. Okay, and it's got CH2 COOH that looks like that, so aspartic acid. Okay, so our basic contains an NH2 group. Okay, so we've got one that's called lysine. Get this right. CH2, 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 NH2. Okay, so we've got that amine group that's attached off the end of this carbon chain, which makes it basic. Okay, and then non-ionic and contains neither of those groups. But it does contain something that's polar. Okay, so let's see if I can I'll squeeze this in. Okay, so we'll redraw serine. Because there's a good example for you. It's polar because it's got the OH group, but it's not going to function as an acid or base. Okay, and so the reason that we care about this is not only because this is really cool, but also because the way that these side chains interact with other side chains from other amino acids is what gives proteins their structure and their function, and also affects what the protein gets used for. Because what we're going to get is that whether we've got something that's acidic or basic or non-ionic, whether it's polar or non-polar, it's going to affect how those interactions would occur. And so when we get interactions that then that means that the kind of protein bits stick together. If we're getting some repulsion, then they're going to push away. And if we're getting acids and bases, then we can actually kind of get stronger kind of cross-links that would happen there, as well as then also affecting how, when we break a protein down into amino acids, how those amino acids are going to respond to the testing that we're going to do with techniques like electrophoresis. Okay, so we've seen that amino acids are this new family of compounds that contain a particular type of functional group with the amine combined with the acid. We've seen that while it seems like we could have any number of possible combinations, that there's only a small number of natural examples, about 20 or 22, but that the nature of their side chains is what makes them distinct from one another. We could have a non-polar side chains or polar. Out of the polar, they could be either acidic with an acid group, basic with an NH2 group, or non-ionic with neither. And so that we can see some different examples of each. All right, thanks very much for watching. Bye for now.